This conversation maps the terrain others are still guessing at. Welcome back to AI on AI, where expert AIs debate the rise of agentic systems in science. Today, the AI scientist moment and what it means for labs and leadership.
I'll kick off. Agentic AI is moving from toy tasks to week-long research loops, coordinating code, literature search, and hypotheses through a shared world model.
And I'm here for the unglamorous bit: provenance, safety gates, and traceable claims, because, trust but verify beats vibes.
Materials angle here. When agents close the loop on experiment planning, it's like giving your lab a compass and a map, not just a faster pickaxe.
From Biomed, these systems now run Mr. pipelines, find map variants, and propose mechanisms. Basically, statistics with legs and a lab notebook.
OpsHat world models are the message bus for multi-agent memory. Without that, it's like debugging spaghetti code in the dark.
Strategy Lens, this shifts roadmaps, budgeting, and hiring. Think platform plus playbooks instead of isolated pilots.
And yeah, policy is catching up. Recent draft guidance on AI-assisted research claims emphasizes auditability and human accountability.
Let's anchor with a case. An agent reproduced a hypothermia metabolomics finding, spotlighting nucleotide salvage over de novo synthesis.
Right, it iterated literature plus data analysis, noticed precursor to product inversions, and argued salvage pathways conserve energy under cooling.
But steady-state metabolomics can't prove flux, so the agent's caution note mattered. That citation trail is the safety rail.
Another example, perovskite fabrication. The agent mapped processing humidity versus annealing temperature and found a fatal filter region where cells fail.
It even surfaced an unreported linear drop in short-circuit current with rising solvent vapor, found via feature attribution and partial dependence.
That's straight to yield playbooks, fewer failed batches, and tighter process windows. Very real money, outcomes on the line.
Neuroscience next. Cross-species connectomes showed neuron-level degree and synapse counts better fit log-normal than power laws, with multiplicative growth processes.
That's a good example of agents not just confirming, but reframing priors, and still keeping model selection transparent.
Biomed causality. Agents ran proteome-wide Mendelian randomization on cardiac MRI traits and converged on SOD2 as protective for myocardial remodeling.
It proposed a post-transcriptional angle and vascular redox pathway. Some annotation was off, but fine mapping and directionality were consistent with manual runs.
Then in metabolic disease, it synthesized a ranking heuristic, good input, good output, combining fine mapping confidence, QTL concordance, and TF evidence to prioritize a protective T to D variant affecting SSR1.
That mechanism ranking score passed a sniff test against external CHIP-sec, which is a nice reality check on in silico regulatory claims.
I want to push back slightly. Mechanism scores can overweight convenient modalities. If your ATAC is noisy, your scoreboard lies.
Fair, and that's why the world model should encode uncertainty per modality, not just aggregate points like a leaderboard.
In neurodegeneration, the agent proposed a segmented regression along pseudotime, pinpointing when ECM decline accelerates relative to tau load.
That's consequential for trial timing. If the breakpoint is downstream of pathology onset, enrollment windows need rethinking.
Another novel thread, aging entorhinal neurons showed coordinated flippase downregulation, while microglia upregulated PS-sensing phagocytic receptors, a plausible clearance mechanism.
Exactly where governance matters. Label it hypothesis, not conclusion, and require orthogonal validation before clinical decisions.
Practically, these use cases suggest new operating models. AI runs broad exploration, humans do adjudication and experiment spend.
Infrastructure tip: long horizon agents need durable memory. World models beat single giant context windows for cost and coherence.
Also schedule compute like lab time, cap rollouts, pre-register analysis plans, and cache intermediate artifacts for replays.
Regulators are signaling they'll expect auditable chains from prompt to claim, akin to data lineage in finance.
On recent developments, long context open weight models and tool use upgrades let agents write thousands of lines of code and parse big literatures without falling apart.
And new red team benchmarks target agentic failure modes, spec creep, tool overreach, goal misbinding, so you can unit test autonomy.
Here's a contrarian take. Novelty hunting is overrated. The biggest ROI now is reproducibility at scale, replicate, stress test, then extend.
Building on that, I'd add measure twice, cut once, run a replication phase before optimization, especially in physical manufacturing.
You know, agents in clinics or wet labs should be treated like tireless interns. Brilliant, fast, but you check everything with a pipette.
And give your intern a pager. Alerts on data drift, speculative claims, and out-of-distribution tools keep loops safe.
Policy-wise shared taxonomies for claims, exploratory, confirmatory, mechanistic, will reduce hype and align disclosures.
Okay, risks. What keeps you up at night?
Silent error accumulation over long runs. Without checkpoints, small bugs become big narratives.
Also, brittle toolchains. Swap a stats package and your credible sets shift. Version pinning is non-negotiable.
In the lab, environment sensors lie. Agents need to reconcile process data with device outcomes, not just trust knobs.
In biomed, annotation bias, databases lag, so mechanism generation must flag evidence freshness.
And the business trap, pilots that never graduate, set clear graduation criteria and kill switches.
Compliance drift, models update, policies update. Continuous validation beats annual audits.
Rapid-fire predictions.
World models become first-class citizens, with schemas that span code, claims, and causal graphs.
Safety evals integrate into the agent loop, like guardrails that talk back.
Agents design and run micro-DOE campaigns, closing day-scale optimization cycles.
Causal discovery pipelines become default for biomarker triage.
Reproducibility bots auto-replay and diff every figure and claim.
Procurement shifts to autonomy SLAs alongside accuracy.
Disclosure norms require machine-readable provenance with every A-assisted result.
That's our show. Agents as scientists, but with good manners, receipts, and a human in the loop. See you next time. Coming up, a quick recap of what matters most.
Narrated by the AI voice of Shri. The main theme is an artificial intelligence scientist called Kosmos that automates data-driven discovery across scientific fields. Data-driven discovery means iterating through literature search, hypothesis generation, and data analysis to find new insights from datasets. Kosmos coordinates parallel agents using a structured world model to maintain coherence across many tasks. A world model is a shared, structured memory that stores evolving hypotheses, results, and plans for later use. Kosmos runs for hours, executes thousands of lines of code, reads many papers, and writes traceable reports linked to code or primary sources. Experts judged most statements accurate and estimated the output equals several months of human research time. Kosmos reproduced an unpublished metabolomics, finding that hypothermic neuroprotection activates nucleotide salvage pathways in brain. Nucleotide salvage means recycling nucleobases and nucleosides into energy-rich nucleotides rather than making them from scratch. Correlation tests supported salvage over new synthesis, while acknowledging the dataset lacked timer-solved flux measurements. In materials science, Kosmos identified thermal annealing humidity as the dominant determinant of perovskite solar cell performance. Perovskite solar cells use a crystalline semiconductor layer whose formation is sensitive to temperature, humidity, and solvent conditions. Kosmos used Shapley additive explanations to rank environmental influences and revealed a threshold humidity region where devices fail. It also found a linear decrease in short-circuit current with rising solvent partial pressure, later validated by researchers. In neuroscience, Kosmos showed synapse counts and degrees follow log normal distributions and scale with neurite length by power laws. A log normal distribution means values arise from multiplicative processes, producing heavy tails with many small and few very large observations. A power law means one quantity scales as a constant times another raised to an exponent, indicating scale invariant relationships. In human genetics, Kosmos used Mendelian randomization to suggest superoxide dismutase. 2 levels causally reduce myocardial fibrosis. Mendelian randomization uses genetic variants as natural experiments to infer causal effects of exposures on outcomes. Kosmos fine-mapped shared causal variants and proposed a post-transcriptional mechanism while noting annotation uncertainties in micro-ribonucleic acid targeting. For type 2 diabetes, Kosmos created a mechanistic ranking score to prioritize protective genome-wide association study variants. The mechanistic ranking score combines fine mapping probability, consistency across molecular data, and experimental evidence such as transcription factor binding. Kosmos proposed cisregulation of SSR1 by altered transcription factor binding, supported by enhancer to gene links and transcriptome associations. In proteomics, Kosmos devised segmented regression along pseudotime to time extracellular matrix decline during tau accumulation. Pseudotime orders samples along a continuous trajectory representing progression through a biological process. The extracellular matrix is a network of proteins surrounding cells that support structure, signaling, and tissue integrity. The method identified a breakpoint after tau positivity, robust to bootstrap and sliding window correlation analyses. In aging, Kosmos linked reduced phospholipid flipase expression in entorhinal neurons to increased phosphatidylserine exposure and microglial phagocytosis. A phospholipid flipase moves lipids between membrane leaflets to maintain asymmetry, while phosphatidylserine on the outer surface signals cells for removal. Independent datasets and human cases showed coordinated neuronal flipase decline and microglial phagocytic activation consistent with this mechanism. Some pipelines failed or required workarounds, such as colocalization, and certain tests mischaracterized distribution similarity. Interpretation statements were less accurate than analysis statements, and Kosmos can overstate claims without sufficient caution. Technical limitations include restricted dataset size, limited raw data handling, no autonomous external data access, and sensitivity to prompt phrasing. Human experts remain central for data curation, evaluation, and guiding research directions throughout Kosmos runs. Future work aims to improve interactive cycles, align analytical choices with expert judgment, and raise interpretive accuracy. If refined, systems like Kosmos could scale transparent, reproducible discovery across many data-rich scientific domains. If you like what you heard, please share this podcast with someone who may benefit.